I follow data. That is what my career is based on, is I follow the data that is interesting. And when I saw these results, not only in cells, also in animals, I was like, okay, I am now a psilocybin researcher because I am going to follow this story for the rest of my life because this is freaking incredible. I mean, I just couldn't believe it. So it's one of those moments. Like, I really can't believe this is what I'm seeing. And I'm just going to have to study this forever now because I want to know everything.
Welcome to the Passing Through podcast, a show dedicated to exploring those spaces not just within, but also between and around us. The cracks in the walls, the moments between the moments. I speak with thinkers, authors, artists, teachers, and everyday humans who have touched the edge of the ordinary and found something to bring back.
What you're hearing are the sounds of mice in a research laboratory. These elderly mice are part of a unique study on aging. In fact, it's the very first study of its kind. At Emory University, a group of elderly mice are about to undergo a bizarre and extraordinary experiment. They're taking psychedelics for science. And what the scientists find out may literally change the way we understand aging forever. Our guest this week is Dr. Louise Hecker, and she's going to tell us all about this incredible story. So instead of a lengthy introduction, I'm just going to take us in. All of the links you'll need to find Dr. Hecker and her study will be in the show notes and on the passingthroughpodcast.com. As always, be good to one another, and I'll talk to you next time. Now, let's turn on, tune in, drop out, and see what happens when you repeatedly administer heroic doses of psilocybin to mice.
Okay, so Dr. Louise Hecker, welcome to the Passing Through podcast.
Thanks so much, Danny. Pleasure to be here.
Yeah, it's nice to sit down with you. I'm glad that we have some time to talk together because your work is at a fascinating intersection of psychedelic research and longevity. And we live in weird times where maybe for the first time in human history, how long we live and what aging is like is being called into question. So I'm excited to speak to you about that stuff. But before we jump into that, I'm wondering if you could tell us a bit about how you wound up here, a little bit about your story and how you found yourself embroiled in this psychedelic research.
Sure. Well, yeah, my research career has taken some twists and turns. Did my training at Michigan, where I was focused on injury repair. And during my postdoc, that's kind of when I got into aging. And so the whole of my professional career since then has really kind of been under this big umbrella of aging. Not so much until obviously recently focused on, longevity science, but really focused on why do the elderly population have worse outcomes with certain diseases. And someone can be totally healthy, can get taken out by a disease in the elderly, whereas the incidence and prevalence of these diseases aren't so high in the younger populations. And so as we age, we have increased incidence and prevalence of certain diseases. And that's really a big part of what my career is focused on, is why is that? And how can we target these pathological mechanisms in the context of aging so that the elderly can, like COVID is a great example, actually, because it's something that impacted everyone. But we all know from epidemiology studies, which is studying populations, that the elderly population had worse outcomes from COVID. And so I don't study COVID per se, but I try to study why do they not recover from COVID, and why do the elderly have increased mortality and worse outcomes from COVID. And so that's kind of the big broad umbrella of what I study. But this project started in a completely unique way, which is a friend of mine who was my workout buddy, Aaron. At the time, he was just obsessed with mushrooms and psychedelic mushrooms, just constantly asking questions about what they do and what's known and what they're in clinical trials for. And he's a lawyer, so he's a smart guy. And I think a lot of my non-science friends are always asking me science questions that, like scientists know everything about science, but don't. And so one day I just decided, well, I'm going to read on PubMed, which is, of course, where scientists get their information from. He's getting his information from podcasts and Netflix, and which is cool. And I like to watch those things too. But I was really interested in, what's scientifically known. So I just decided to start reading, and then I thought, well, I'll just get some factoids that I can go to my workout and surprise my friend with some interesting facts. And I just started reading and I went down the rabbit hole. I just was really fascinated by psilocybin. It's such a cool drug. I was so intrigued by how many different diseases it was being used for. And one of the things that struck me was that people were reporting that just one dose led to positive impacts up to 5 years later. And to me, that didn't make sense because, I mean, the drug's out of your system in 24 hours. So, how is that possible? And that kind of got me thinking. And then I came across this idea that someone had put out there that, maybe one of the ways that it's working long term is that it's doing something to impact aging. And I just found that idea really intriguing. And I thought, well, I study aging. I know exactly how to test this. And that's kind of where it all began.
Okay. So then you had the intention. It wasn't like this study was some accidental discovery where you were looking into something else and said, "Oh my God, this is de-aging mice." You went into it with the intention of, is there a correlation or is there some link between psilocybin and aging?
Right. It was a completely off the wall. And this is kind of, I'm an outside the box scientist. And I love, just random projects, especially, I'm a curious person. I think all scientists are just curious people, right? And I am an especially curious person. And I just thought, wow, this is really easy to test. And it's just such an intriguing idea. Why not test it? And so I thought this was going to be real easy study. We're going to order the compound and we'll start experiments next week. Turns out that's not what happened at all. um, I had no idea going into this project how challenging this would actually be because psilocybin is, as you probably know, is a Schedule 1 drug. So it's in the same class as heroin.
Mm-hmm.
And that makes it extremely challenging to do research on. So, normally if I'm interested in a drug or a compound, I can just order it and we can start experiments the next week. That's what I thought was going to happen. But turns out you need a DEA license. You need a lot of regulatory things in place to even bring that into the lab. And so I thought, okay, well, I was, I think, blissfully ignorant of like what that process would entail at the time. Because had I known, maybe I wouldn't have gone down this path. I don't know. But long
That's
story short,
actually
yeah, go ahead.
That's actually an interesting point. How do you feel about that now? Like, do you feel like, because when I hear you say that, I feel like that could be a deterrent for breakthrough discoveries. And if it's an obstacle for science to just get access to this substance, how does that feel for you?
Yeah, I mean, I could not agree more. It is a huge obstacle. And some unpublished work that we have right now is looking at a systematic review. So, we basically found every paper in the US ever published on psilocybin, and there are only a few hundred. And
Hmm.
that's like from my standpoint, that's crazy considering how many different clinical trials it's been in, how long it's been around as a frame of reference, like resveratrol, which is like a compound that's, reduces oxidative stress. It's just a supplement, and it has 20,000 papers, right? So it's, you're right. And that the Schedule 1 status of psilocybin has made this something that's very challenging and difficult to actually study. I certainly was not aware or had any understanding of what that meant or what that looked like, but now having gone through the process, I can see that, yeah, it's a big problem actually in terms of making research progress, a big challenge.
Yeah, yeah. It's nice to hear that that sentiment is present inside of science, because to me it's baffling just on a philosophical perspective, where it's like this is a fungus that just it just grows. Like how is this a Schedule 1 substance?
It's beyond me. I think that things are moving. I liked the direction that things are moving to. Like, it seems like there are changes in the government that are happening right now with new, executive orders to expedite review of psychedelic
Hmm.
therapies, clinical trials. The thing that makes so that the conditions that make something a Schedule 1 drug is it has no accepted medical use, it's questionable safety profile, and potential for abuse, those 3 things. So I think the potential for abuse, I guess we can't really remove that, right? It's a drug, people use it recreationally. But at the same time, no one's ever died of a psilocybin overdose. There have been incidents where people have taken psilocybin and then done other things, but no one's actually died from too much psilocybin, right? So it's obviously been in many clinical trials. So I think the safety profile is great, is very good from everything that we know. And we know a lot about the safety profile. If it gets approved for any disease indication, which I know it's in phase 3 clinical trials now. So if it does get FDA approved for any indication, that will give psilocybin an, quote unquote, accepted medical use. And so I think once that happens, that will lead to a change in its Schedule 1 status. So I'm really hopeful that those changes are kind of coming down the road. It will really open the door for others like myself that are interested in studying and make it easier to study. So yeah.
Cool. That's good to hear. And I like the optimism. And yeah, I want to move forward now back into the study, because this was talking about the hurdles that you were facing to actually conduct this study. But
Mhm.
let's talk about the specifics of the study.
So once we got psilocybin into the lab, which by the way took over a year of getting all the regulatory things into place, getting a DEA license, getting the controls that you need in order to actually study it and possess this in the lab. And so once we got into lab, the first experiment, which I had already planned a year prior, was really simple actually. It was to well, actually, let me back up. So psilocybin, as you know, is the psychedelic compound, the active compound in psychedelic mushrooms that causes you to hallucinate. Now, when you ingest psilocybin, it's in your body, it's broken down into psilocin, which is the active metabolite. So psilocin is what your cells see systemically, and that's the active metabolite. So we gave cells psilocin or vehicle, so the carrier, vehicle control. And basically, we have these cellular models of aging. So cells, like people, they age and they have a natural cellular lifespan. And so what we can do is watch that cellular lifespan over time with or without psilocin. And that was the experiment, really simple. And so, I was hoping to see that there was some kind of impact, but I was just absolutely astounded when I saw this really striking dose dependent impact, meaning the more psilocin you gave, the more life extension the cells had. So, up to over 50% life extension. It was very reproducible and it was dose dependent. So we knew right away from experiment 1 that we're really onto something.
And what types of cells were these? These were human cells?
So a lot of what I study is fibrosis, which is the clinical term for scar tissue. Like if you think about, like if you fall scrape your knee, you have a wound, like your first line of defense is inflammation, but your next line of defense is your fibroblasts, which are kind of dormant until something happens. And then when you have an injury, like you fall and scrape your knee, they become activated, they proliferate, they migrate to the site of damage, and then they actually contract out cells. So that's actually what helps to pull the wound closed and secrete the matrix, which is the tough part of the scar. And then as wound healing is culminating, the cells undergo programmed cell death, apoptosis, and you have resolution of your injury and healing. So fibroblasts are a good model to use for things like these cellular replicate. We call it replicative senescence models, where you just simply watch the cells divide over time until they become senescent, or that's the term for the cellular term for old. They stop dividing, they stop working as well. As there, you see a lot of changes when they become senescent, and so because I had fibroblasts in hand, and because they're a well-validated and characterized model of cellular aging, we used fibroblasts. And so initially, now I study a lot of lung disease. So I had lung fibroblasts. And we've also repeated these studies in skin fibroblasts. So two different types of fibroblasts from different parts of the body. But again, most research to date has really focused on what psilocybin does in the brain, and probably only a handful of studies have ever really looked at what psilocybin is doing outside the brain. So I was really focused on using a cell type that was outside the brain.
Yeah, that's fascinating. I love this concept that psilocybin and potentially other psychedelic substances aren't just having their area of impact be on the brain, that it's
Right?
actually systemically affecting the body. Because is it true that most cells, or is it all cells, I don't know, have these kind of receptor sites where they can interact with psilocin?
Yeah, that's correct. And yeah, it's funny because again, I had never studied psychedelics, never studied psilocybin. So I really, sometimes it's actually a really good thing to walk into a project not knowing anything, not having any idea of how things are going to go. You're totally open to, how does this work? You don't have any preconceived ideas of what's going on. And so in some ways it was kind of good. I was kind of like, whoa, what is happening? And you're right, actually. And that's one of the things I learned because I didn't know anything about psilocybin and how it's and signaling. And so I learned that psilocybin signals through the serotonin receptor, which surprising to me is because when I think serotonin, I think brain, you
Hmm.
know, oh, that's just the first thing. So I had no idea. It was actually a surprise to me that serotonin receptors, yeah, are pretty much in almost every cell type. And so at some levels, it's not surprising, right? Like if all of your cell types have this receptor, why? And why wouldn't it impact other cell types, right? We just didn't know how, or if it does, we don't have any evidence that it does. So yeah, so to your point, yeah, that serotonin receptor is on most cells in your body. And we've since found, since the paper, we found that yes, it is acting through the serotonin receptor. Some of our unpublished studies have also identified other receptors that seem to be important in this longevity signal process. So we're really kind of drilling down on that now, is exactly how it's working is certainly through some of the receptors we would expect, but also through receptors we may not have ever expected.
And maybe we could talk a little bit about that in a bit, because when I was reading this study, and I'm a layperson, I'm not a scientist, so when I'm reading it, the things that are jumping out to me that are surprising are things like the physical characteristics that were observable and measurable that you could see. You don't even need a microscope, because I guess maybe I'm jumping ahead a little bit, because the next part was, I guess, bringing this to mice, right?
Yeah, well, let me walk you through what we saw in cells first.
Okay.
All of that is really kind of what led us, right? And I will also mention too that one of the challenges, along with regulatory things that you need to get psilocybin in lab, is that funding is a challenge, right? Because as a scientist, right, I work on projects that I'm funded or contracted to work on. So the way it works is, I have this idea, here's my data, I submit these things to get funding, and then I'm then funded for X amount of years to work on X, Y, and Z, right? So we always have kind of slush funds and additional funds to kind of work on these kind of risky new projects that aren't funded. And that's how we build evidence to get the bigger funding, right? But I want to point out too that we didn't have funding to work on this, right? Like this is just a crazy side project I decided to work on. And not only that, it was a crazy side project that everyone else thought was crazy. So no one would fund this project. At the time I was working at Emory University, and Emory had given me a pilot fund of $50,000 at the time to do these cellular studies. And so that's not a whole lot of money to do research with, right? Compared to
Recreationally, it's
that we were working on. But $50,000 is enough to like do these initial studies to see if there's a story. And so I was really grateful that they had given us this funding to do this, right? So we found the initial finding, it clearly has an impact on cellular lifespan. So then we started looking at, well, how is this working in the cells? And the first thing I did was to look at basically every aging marker that I had in the refrigerator in terms of antibodies to evaluate like what's happening inside the cells. And to my surprise, every single thing that I looked at was rescued to some extent by psilocybin in a dose dependent manner. So DNA repair mechanism, markers of DNA repair seem to be working better, less oxidative stress. So oxidative stress is something that accumulates with age, and the psilocybin treated cells had oxidative stress levels that were similar to young cells. I mean, so this typical increase in oxidative stress that happens just did not happen in the psilocin treated cells. We saw markers of cell arrest, which again happened with aging. And at some point, the cells did become aged and senescent like normal cells. It just took them longer to get there. So
and
they
and
had
how
a
much longer? Like how much longer are we talking?
Yeah. So, well, cellular lifespan, like typically, and this is a range. So I don't want to say like it's a number because it depends on the cells and where you start the cells. But typically we count population doublings, which is how many times the population of cells doubles, right? That's how we know how much they are replicating. And typically it can take 40 to 60 days in continuous culture of replicating. We count how many times literally the cells have divided over the course of their lifespan. So typically this process where they peak out and become aged takes around somewhere around 40 plus days. Psilocin treated cells, depending on the dose, was living to like 90 plus days at times, depending on the conditions. So it was clearly a huge dramatic increase and essentially doubling their cellular lifespan.
So just because that's a staggering number, when you saw that, what's going through your head? What was that moment like when you're like, what the fuck?
Yeah, basically, yeah. I mean, it was shocking, but it was we again like we knew this was really fascinating from that first study, right? We knew because it was just so obvious that there was a dramatic impact, right? So then, looking to see, well, how the heck is this working? And then the second surprising thing was, again, I study aging, so I have tons of antibodies to look at markers of aging, not specifically for this project, but for all of my age-related projects. And it was surprising to me that I thought, well, let's test out everything I have, which is things related to DNA damage, related to proliferation, related to oxidative stress. And I was expecting like, okay, this will give us a better idea of what's happening in the big picture. I didn't expect that every single marker was going to show some kind of rescue from psilocin. So that was the next big surprise that everything I looked at. I mean, I don't have projects like that it's like, well, okay, maybe it's just oxidative stress or maybe it's working through this, but it was like everything. So it opened up more questions basically. It didn't give me like a defined path. It was like, it's working on everything related to aging, everything I'm looking at so far. Then we looked at telomere length. So telomeres are protective caps on the ends of your DNA. And another analogy I like to use is like the plastic tips on the end of your shoelace. And when you buy those shoes, they're like nice and crisp, and those caps look pretty and defined. But as the shoes wear and tear, you can see like those caps are starting to fray and shorten. And that's essentially what happens to your telomeres. And so over time, you have this reduction in your telomere length. And that is a really well validated biological marker of aging. So as we age, your telomere becomes shorter and you have less protection of your DNA because of that. And that leads to all kinds of, age related things. So, less protection of your DNA, as you can imagine, causes all kinds of impacts in your cells.
And that, like, just for a person listening, the kinds of things that might cause are like increased susceptibility to certain illnesses or like cancers or cell mutations or
DNA damage, like, less protection your DNA has, more it's susceptible to all types of things, mutations, damage, less proliferative capacity. A lot of things kind of coincide with that age dependent shortening of those telomeres. And to our surprise, psilocin treated cells, their telomere lengths were preserved or long. So they weren't shortening like the same as the cells that were treated with a vehicle that, we normally see this reproducible shortening of those telomeres. We did not see that to the same extent in psilocin treated cells, suggesting that you have more protection of your DNA for a longer period of time. So that was really fascinating. So yeah, from these really exciting findings, and then, again, we repeated this in another cell type, and we, so we did all that we could in cells, and we were like absolutely convinced that we have to do this in animals. And so I was able to get another round of pilot funding from Emory through this mechanism called the WOW Award. And it was perfect for me because it's a internal pilot funding for high risk, high reward projects. And I was like, well, this is it. Yes. I mean, and by the way, I was submitting grants and they were just getting trashed. Just that this idea and concept was just like not ready for prime time. No one thought the data was compelling enough to fund it, or that just maybe the priorities of these funding agencies wasn't aligned. But yeah, couldn't get funding. So once again, Emory came through with some pilot funding that helped me get to the next step.
is that a thing? Is it stigmatized? Is this kind of research seen as like, oh, yeah, like it's hand wavy, and so people don't want to fund it? Is that where this comes from?
Well, I think I'm getting hit from two angles, right? One is, yeah, there's a huge stigma around psilocybin for whatever reason. Like, look around and you can see why there's a stigma. There's also the fact that it's a Schedule 1 drug, and when I would submit grants, believe it or not, one of the criticisms is it's a Schedule 1 drug, and it's like, well, no kidding. Thanks for letting me know. I have a whole page about the fact that it's a Schedule 1 drug and why it's likely to be reclassified down the road. And also, just because it's a Schedule 1 drug doesn't mean that it doesn't have value. We shouldn't be testing it.
Yeah, of course.
So, of course, I don't agree with that being a criticism, but, those are the criticisms I would get. So part of it is just government priorities, because at the time, submitting to government agencies, just didn't feel like this was a priority, as well as maybe there wasn't enough research, or it was almost kind of an unbelievable idea and then when you're not published and it's just preliminary data, you don't have what I call street cred. Publications are your street cred, right? Because then it's been peer reviewed, other scientists agree, it's published, and then you have some credit. But until then, it's just some crazy idea by Dr. Hecker. So that's one part. The other part, I think, is that longevity science is also tougher to get funding for. So people want to fund, or agencies tend to fund more like disease specific focus. But aging's not a disease, right? So people in the longevity space sometimes, have trouble getting funding for this, believe it or not, because it's not disease focused and there's just less funding allocated toward straight up longevity science. So, kind of getting hit from two angles did not make it easy to get funding at the time. That said, yeah, I got my next round of funding from Emory, which was $300,000. And I knew that, again, that's not a lot of funding for a big research project, specifically an aging research project, which is very expensive. But I knew I had one shot. I was going to take that shot and I was going to make it the best experiment that I could design. And so we spent a lot of time figuring out how to design this experiment, right? Because aged mice are very expensive. They're like
Hmm.
$500 a piece, believe it or not. So just to purchase mice to do an aged mouse study is a tremendous expense, right? So
Were they more expensive than actually purchasing the psilocybin? Ugh, I can't keep
yeah,
saying it wrong.
they're both expensive, to be honest. They're both a lot of people ask me like, "Oh, but they're just mushrooms." And I'm like, "No, I can't use just mushrooms. I have to use pure medical grade psilocybin from a DEA approved vendor. It's not like I can go down the street and pick some psilocybin mushrooms." These are very controlled experiments. Of course, we need to use pure medical grade psilocybin. So yeah, there are a lot of expenses for this kind of study, but what we wanted to design was a realistic study. If you think about it, okay, if psilocybin really does have this, what I call geroprotection, it protects from aging, then what's a realistic in the real world? It's not realistic for someone to start taking psilocybin when they're born or when they're 10 for the rest of their life. That's not going to happen. Right. So
That's what I did.
yeah. Right. Yeah, I mean, if and maybe, people are certainly, testing it out recreationally, but, as far as like kind of a therapeutic benefits, when does it really matter and when are people really gonna need it, use it? When is it gonna, when is the best time to actually evaluate it for its geroprotection? And so we thought we've got to design a realistic study. And so we started with aged mice that were 19 months of age. That's like the equivalent of like a 60 to 65-year-old person. And the reason we wanted to start with aged mice is because, that's the exact age right around your 60s is really the time when people are starting to experience really dramatic kind of age dependent shifts in their health. You're more susceptible to things. You have more incidence of things. So, I mean, I study a rare disease of pulmonary fibrosis in the lungs, and the average age of diagnosis is 66. So, studying diseases of aging for my whole career, I really wanted to think about this in the context of age-related disease and what it could do.
Hmm.
So yeah, so we started with aged mice because we felt like that was more realistic. Then we had a lot of discussion over like how we're going to do dosing. There's so much out there, right? There's microdosing, there's hallucinogenic dosing. Now, we wanted to use the hallucinogenic dose, I think, one, because we wanted to see an effect, right? So if microdosing works, that's something we could look into. But right now, my primary goal is really what's a dose that I'm going to see an effect. And there are just more research and data on hallucinogenic dosing. There's a lot less known about microdosing from a scientific standpoint. We basically looked at every single study ever done in mice and every single clinical trial ever done in patients. And we kind of gathered all of that information and we came to one protocol. And I've certainly received some criticisms on the protocol and the dose, but it is what it is, and it was a starting point.
The
Uh,
the the just to see the criticisms, because for me, the only thing I could anticipate is people who would be concerned about the ethical implications of just dosing these elderly mice. Is this where the criticism was coming from, or was it somewhere else?
it's funny that you mentioned that's not what I was talking about. I'll get to my point, but to your point, this was a crazy like talk about regulatory things that make research challenging. I mean, to get this study approved, because when you do an animal study, you have to get IACUC approval, which is, IACUC oversees all animal studies in academic institutions. So this went all the way up to like the ethics committees. And I mean, there was a lot of pushback on my experimental design because one, it's the equivalent of saying, like, I'm going to give mice high dose heroin for the rest of their lives. And it's never been done. And repeated dosing in mice has never been done. And it's certainly never been done in the context of aging. So there were all kinds of safety concerns, right? Ethical concerns. There were concerns of how we're going to protect from people stealing the drug during the experiment, right? And how we're going to code things. I mean, so yeah, it took a lot in terms of figuring out and getting approval. As one example, so my original proposal was to study lifespan, like, does it increase lifespan? The ICOOC would not approve that study because they said, one, there's never been a study in mice giving hallucinogenic doses repeatedly, never been done in the context of aging. So the safety issues were a big concern. And, and I understand that as a scientist, that makes sense, right? Like there is a concern that's never been done when something's never been done, so we had a lot of strict monitoring and things that we had to do to check the mice and to safety of monitoring these mice regularly, right? So, but they wouldn't approve the life extension study, but they would approve a study where we could go to 50% mortality. So when any group hit 50% mortality, so median survival, that signals the end of my study. And so we didn't know how long this study was going to go, but we knew that whenever mice hit 50% mortality in any group, that's when we have to stop. So that's another example of like how the regulatory things kind of dictate, in some sense, the design of the study because I couldn't actually do the study I wanted to do. But they said, well, you should have an answer and you'll know a lot more about safety in this context by this initial study. So we said, okay, we'll do that. And we arrived at the dosing of once a month in the mice, which is probably more in line with like once a year in humans. And the actual dose we gave was again, we knew it was a hallucinogenic dose in mice because we knew from other mouse studies that these doses elicit hallucinations in mice. So we know that mice are having hallucinations because they have this what's called the head twitch response. So we actually had like GoPro cameras above the mice monitoring to ensure that they had this. And so when they give the drug, that you can actually see like every once in a while they have this like little head twitchy thing that happens that doesn't happen in the control mice. And so that's indicative of that the mice are hallucinating.
An interesting, because I hadn't thought of this, like I hadn't put myself into the actual headspace of you like doing this research. Would you actually be there? So the dose is administered to the mice, and you have to sit there and watch them. And watch what's happening. You have to look for these ticks and see their behavior. What do mice do when they're on mushrooms? Do they like play some music? They're dancing. Like what are they actually doing?
I mean, this was so fascinating to me that like the first day we did the dosing, it was like Christmas morning to me. I was like so excited. I was so curious about what was going to happen. I had no idea. We knew that the head twitch response was indicative of that they are having hallucinations. So we had GoPro cameras on top so that we could quantify that if we needed to, just to show evidence that they were, like somebody raised their hand in one of my talks said, "Were the mice tripping?" And I was like, "Yes, they were." Because we can actually quantify that they're having this response. But yeah, so I was super curious, what are the mice going to do? And they have this response, and then it's very subtle. I mean, you can just see like they're kind of every once in a while kind of shaking their head, or once in a while they kind of like bat their tail a little bit more than often than you don't really see in the control mice. But within like half an hour, the mice just kind of go to sleep. It was anticlimactic. Like I was expecting them to be kind of like looking around, like, whoa, but no. They just kind of get tired and sleepy, and then they kind of just go to sleep. The drug is pretty fast acting, so they're pretty much back to normal within a couple of hours.
Yeah. And I guess another question that came to me is compared to a human, because even in a human, there's a psychedelic dose, but there is a whole range of psychedelic doses. You could go up from mild psychedelic dose to a heroic psychedelic dose to like, I need to go to the hospital dose where I'm like, I can't function anymore. And for a mouse, do you have a sense of where on that spectrum it is?
Yeah. So yeah, it's hard to say exactly, but I mean, for sure they're in like heroic dose range, and that was the goal. Because we wanted to, again, because I think scientifically there's a lot more known about sort of high hallucinogenic dosing in humans, as well as the impacts in humans, right? So I wanted to stick with that because that's really where the data is from clinical data. So I wanted to keep in line with what's already known out there. There's a lot of factors though, because people keep asking, like, how is, and even like within that, like you said, within that high hallucinogenic dose, there's still a huge range, right? Even in clinical trials, there's a massive range. So we knew we wanted to get it within that range. Now, there's a lot of differences between mice and humans, of course. One being, and this is one that I factored in heavily, is that mouse metabolism is about 4 times faster than human metabolism, right? So we did factor things like that in. I know it was a pretty high dose, and I know it was a hallucinogenic dose because of the data that we have. But yeah, it's kind of questionable in terms of like how to relate that to human trials. But I would, again, this is that area since the paper came out has gotten some like criticism, like that's too high of a dose compared to clinical trials. And I disagree, obviously, because I think there are things that people are not factoring in, like mouse metabolism versus human metabolism and things like that. Again, we had like our best minds looking at every clinical study, every mouse study, and kind of putting this protocol together. And then what we arrived at is the dosing we put in the paper. We did give one kind of lower acclimation dose initially, just to ensure like that we didn't see any adverse effects. Again, it's never been given in the context of aging, so we really had no idea if there would be adverse effects. So we gave an acclimation dose, a lower dose first, and then that dose was eventually the next dose was triple that dose, and we gave it once a month for as long as it took, which turned out to be 10 months because that's when the control mice hit their median survival.
Okay.
So yeah, certainly a lot needs to be done with in the future with dosing, and I can tell you some of the ongoing studies we have now with microdosing and other things after I get through this piece. But yeah, so we gave them high heroic dose once a month, and turned out to be 10 months because that's when the control mice hit 50% mortality. And now, really early into the study, within a few months, one thing that everyone in the lab noticed is that the psilocybin treated mice looked better, just physically looked better. And it wasn't even something I could like tell you how or why at the time because it was kind of early in the study, but they did just look better. And we all noticed it and we kind of kept our eye on it. And then as time went on and we saw that the vehicle treated mice were dying and the psilocybin mice were not dying as fast, it was pretty obvious, right? So in the study we published that when vehicle hit 50% mortality, there was still 80% survival in the psilocybin treated mice, which was a very dramatic increase in the survival benefit. But at the end, and again, because I didn't really know at the time how to quantify like how they looked better, aside from photographic evidence, and I don't study, I don't study longevity. I don't study. So all of this was kind of like, well, how do I quantify that, how they look better? But if you look in the paper, you can just see that the aged mice, like humans, like some have a belly, some have a little hunchback, some they're losing hair and getting bald spots, they're getting white hair. That's typical aging in mice as well.
I have all of those.
Yeah. Yeah. And we see that in mice, and it's very typical, and it's, just like people, it varies from mouse to mouse. Like this mouse might have like a lot of white hair, and this mouse might not have much white hair at all, right? Just like people. So we're seeing those typical changes with age in the control mice, but just the psilocybin mice not only looked better, had less of these things like the belly and the hunched back, but just their hair quality looked shinier and nicer, whereas the aged mice had kind of like scruffier, less fluffy, shiny hair. An interesting thing was that some of the again, because the mice were like equivalent of age 60 when we started the experiment, so they are already had some phenotypic changes that are associated with aging, like bald spots and white hair. And one thing that was just like shocking to all of us was like the bald spots on many of the psilocybin treatments grew back hair, and the white hair grew back in black. I've never seen anything like that. So that was just like, whoa, one of those whoa moments.
Yeah, like that's so fascinating to me. When you're seeing that again, it's one of those moments of, okay, how can this be? Could there be any other explanation for this? Like, I'm sure you were having these thoughts, and the place that you arrived was, it's this. Like, there is no, at the moment, there's no other explanation for how this is happening except that the psilocybin is.
Yeah. I mean, like, as a scientist, I mean, the only thing that matters to me is the data. I follow the data, I've had examples of projects where, I did a project and it was going, and I completed kind of what I wanted to do, but I didn't want to keep following that project either because it wasn't exciting to me, or some of the angles I chose to take didn't pan out. My hypothesis was wrong, or I just had, other projects that were more interesting or whatnot. I mean, the bottom line is like I follow data. That is what my career is based on, is I follow the data that is interesting. And when I saw these results, not only in cells, also in animals, I was like, okay, I am now a psilocybin researcher because I am going to follow this story for the rest of my life because this is freaking incredible. I mean, I just couldn't believe it. So it's one of those moments, like, I really can't believe this is what I'm seeing, and I'm just gonna have to study this forever now because I wanna know everything.
Yeah, yeah, and I would too. That's it's unbelievable to me. I almost wish I was there to see that happening. It's like something out of a movie. And
Mm.
and I wonder, did you also study what the cognitive state of the mice were? Did you see improvements in their cognition or their memory, or was this purely physical changes?
Yeah, great question. So, before I answer that question, I'll say that with these findings, then I was like, "Okay, now I'm ready to get grant funding because I have some really exciting stuff." And the reality is, for me to continue studying it, I need more funding because I've used up my pilot funding in the best way possible and gotten this incredible data with that funding. But in order to understand more, I'm going to need bigger funding. So at that point, I started submitting grants like crazy. Nothing hit at the time, nothing, right? So I
Wow.
still, and I was like, I can't believe with this data in hand, like nobody is gonna fund this. I couldn't understand. And so at that time, I decided that even though we have a lot more data that we are collecting and, continued going, that I needed to publish the finding itself. Because if I don't have, like I said, my street cred, then I'm never going to get more funding to continue studying this, right? I need to have this go through the scientific, rigorous review process for people to believe the finding itself that this is impacting aging. And so we kind of like put a cutoff point in terms of, okay, this is the finding in cells and animals that it slows aging. And that's where we published the paper. And it was a tough paper to get accepted too, by the way. It was like a lot of, I'm sure the journal was probably like freaking out, like this is a really sensitive topic. So they had like a lot of things that had to be addressed, and from a scientific standpoint, which of course we did in published paper. That said, now, and I can talk about what's come after that with funding, but yeah, we did collect. So from that study, from that initial study, and we published on the high dose, we collected everything from brain function. We looked at spatial learning and memory. We took all of the organs and the blood. We did transcriptomic profiling, proteomic profiling, metabolomic profiling, and we just took everything that we could get because we just thought, this is a one-time study. Right now, it's the only money I have to do is for this study. I'm gonna take every single piece of data that I can from it. So we did some quick things like test whether it's impacting the brain. We haven't published on any of this yet, but yeah, we are finding that in those mice that had, and you'll see this data coming out in publications hopefully soon, that the mice had, probably not surprising, right? Based on what's known. So there's so much known about what psilocybin does in the brain. Wasn't really surprising to me that they had improved learning and memory. And actually, we in fact looked at the brain itself, and the brain had less atrophy compared
Mm.
to normal aging mouse. So there are a lot of really interesting impacts in the brain. My interest, of course, is really less focused on the brain, because if there's one thing that we know a lot about is like what psilocybin does in the brain. What we know absolutely nothing about is what it does in the other organs. And so we are putting together our findings right now to submit for publication on all of the omics. And it is fascinating. Like, I can give you like a sneak peek of one thing that I think is really interesting, which is that I, of course, would have thought that the brain is the organ that has the most protection. And based on our profiling data, actually psilocybin has greater protection in other organs from like a transcriptomic profiling
And
standpoint.
what does transcriptomic mean?
So like what we did is we can do bulk RNA-seq, which should tell us the gene expression patterns of all genes in every organ. And then what we can do is, okay, let's say if we look at the brain, right? We look at a transcriptomic profiling of the young brain versus the old brain, and then that defines a signature of, these are the pathways that are altered in aging, and we can actually, with bioinformatic approaches, validate that based on publicly available data sets from other studies, even. So we can first validate, like, yeah, our transcriptomic profiling changes in genes, in every gene in that organ. These are what happens with aging, and that can be validated, from other studies. And then we can look at, relative to that, what the aging brain versus the aging brain plus psilocybin changes are, and look and compare that to, okay, this is a typical aging brain signature. This is a psilocybin brain signature. And how does that look relative to young?
Mm.
And we can say, okay, yeah, actually, the aging psilocybin brain has more pathways in common with the young brain than with the aging brain, right? So we can look from gene signatures of what are typically changed with age and how that is altered in response to psilocybin, right? So we've done that with every organ, and we can show you how, from a transcriptomic profiling standpoint, the gene expression patterns are more similar to young, or we can show you exactly which pathways and genes expressed are altered in aging and in response to psilocybin, and then even compare that. And what we're seeing is that, yeah, psilocybin has the potential in all of the organs that we've looked at to lead to gene expression patterns that are more similar to young than with aging.
That's super fascinating stuff. I mean, it tracks when I think of any of the times that I've personally ingested psilocybin. It's not something that feels like it's only affecting the cells in the brain. It feels like every cell in the body is affected, and your research seems to confirm this. And
Oh,
I
yeah.
wonder, did you find anything unique about the cells in, say, the stomach? Because the stomach and the digestive system, when you consume psilocybin, is particularly strangely affected. You know, they say that the stomach is the second brain and that there's a lot of serotonin in the stomach. So I would guess that there might be some weird things about the digestive system. Did you see
Yeah.
anything?
That's actually because, again, this was like all based on very limited pilot funding. We sort of had to like pick and choose. I wish we had looked at the stomach, but we didn't. We looked at
Okay.
the brain, the liver, the kidney, muscle tissue, the lungs, skin. So we kind of pick and choose, but certainly like with new funding and new studies coming down the road, we will be looking at absolutely everything. So
Cool.
yeah, yeah,
And the stuff that you did look at, is there any one thing that surprised you most where you were like, wow, the stuff that's coming out of this organ or
yeah.
this part of the research is more surprising than others?
Yeah, I think the one thing that really surprised me was from a transcriptomic, proteomic, metabolomic profiling standpoint, the lung seems to have the most protection, which was extremely shocking to me because, well, first of all, I just expected the brain would have the most protection because of what, we already know and because it causes psychedelic effects and just profound changes in the brain that we already know about. So surprisingly, other organs are equally and more affected in terms of their kind of signatures of gene expression, protein expression patterns being kind of reverted back to young seem to happen more. Of course, it happens in the brain for sure, and to a large extent, but in some other organs, like the lungs, for example, had the greatest protection from that standpoint, which makes me think that, there may be some benefits to, psilocybin for lung diseases and other diseases for other organs. The kidneys seem to have less protection than all the other organs for some reason. We don't know why yet. One of the things that we're also studying now is, of course, we're most interested in the benefits, but we also are interested in the potential harms, right? No one's ever taken, even in clinical studies, they're, I'm not aware of one off the top of my head, at least, where there's been repetitive psychedelic dosing over a long period of time. Usually it's like once or twice, sometimes three times, but never over a period of years or, so we don't know anything actually about repeated psilocybin dosing. And that's something that we do have to keep an eye on because, again, nothing's known. And so whereas there, just like any drug, right? Like if you take aspirin, if you take half an aspirin, you're not going to have any effect. If you take two aspirin, three, you're going to have a beneficial effect. If you take 100, you're going to die, right? So like any drug, there are kind of these optimal levels of you're not going to have an effect to optimal effect to detrimental effects. And so that's something that we're really closely studying now is to sort of understand that range and what leads to the most beneficial effects without causing any detrimental effects. And that's really important in terms of, people keep asking me about the translation and, when it's, but it's not so simple because there just needs more work and studies for us to really define that safety profile for its use as a geroprotective agent.
Hmm. Yeah, thanks for bringing that up because that was a question I had too about not just maybe necessarily the safety, but also was there any negative things that you noticed? Because the stuff we've been talking about so far is overwhelmingly positive and astonishing, and I wonder if there's anything in there where you noticed harm.
Right. I mean, we didn't. I can't say like there's this one thing. I would say I'm going to keep a closer eye on kidney function moving
Hmm.
forward. Not that we noticed anything detrimental, but because it had so much less protection than all of the other organs that we looked at, I'm interested in really following to see, well, is there any, and it did have some protection, by the way, but it just really wasn't even close to like all the other organs had really incredible level of protection from the omics data that we've looked at. But to answer your question, I didn't notice anything overt that makes me concerned, but there are certain things that I'm keeping my eye a little closer, look at things moving forward to specifically monitor and to think about, potential harms. Things like cancer risk, that's something that nobody's thought about. Now, I'll say like in cells, there was no evidence of that, right? Because cells can become transformed and turn cancerous. By definition, they have uncontrolled proliferation, right? And we didn't see any of that, right? At any dose, at any level. So cells will get old and they will eventually stop dividing. There was no evidence of oncogenic transformation. We didn't see, I mean, we didn't do a quantitative study where we were looking at tumors and things in mice because, the bottom line is even when we started, mice probably have some like tumors because they're already sick in their, equivalent to 60s. And we do see in aged mice, we see like different things like, increased tumors and things like that happen with aging. We didn't see any overt differences in psilocybin treated mice versus control mice. But that said, we weren't doing a quantitative study to look at cancer risk, right? So those things do need to happen and we are working on those kinds of things.
Yeah, that was going to be because I guess now we're heading into the final section of our discussion, which is like, what does this mean? What happens next? How do we go forward? And it sounds like some things you're already thinking of are potential harm, safety, more targeted data on like cancers and things like this. Are there other things that you're interested in pursuing?
Yeah, gosh, well, like I said, now I am a full-blown psilocybin researcher because this is a major focus of my lab now. And I was really torn about whether to put the paper out because it was a very short paper that just kind of had the finding and it, we have all this other data that I would have loved to include in one huge paper, but I just thought I'm never going to get more funding if I don't put the finding out and to keep going, right? So at the same time, I thought, this is so exciting. I want to start presenting it and want to get it out into the world. Yeah. So what are we doing now? Well, it's just, the best part about this was getting that paper out because it created it created this crazy viral explosion of attention, which I never thought would be important in research because it's like I'm doing research to do the research, not because of any, but what it actually, I never realized what a benefit that would actually be because now it's like in the public domain, people's eyes are everywhere. I mean, it's like every time I look on Instagram, there's like a new post about our data, there's a new news story. I mean, I think there have been like 150 news outlets that have reported on it. And the real amazing benefit of all that is that now it's been out there in the world. People are contacting me.
Mm.
Can we work with you on this? Like we have experts that we're working with on the signaling aspects, right? So like blocking receptors and seeing, because certain receptors may be important for psilocybin for its hallucinogenic impacts and for other certain impacts. But for the longevity signal, we're working with some really cool people that are helping us to define the signaling pathways. And we're finding that it actually is signaling through not just the typical serotonin receptor, but other receptors that are playing a role in this longevity signal. So, from people like that to that, we're now working with the Vail Health Foundation to do the first clinical trial in healthy aged individuals with psilocybin, and the primary endpoint is aging. And that funding and that project and all of this really transpired because the paper came out, right? And so that's super exciting. So that clinical trial is being planned, and I've been part of that planning, and that's an exciting, career milestone for me is to have, hey, something I worked on and discovered is now going to be in clinical trials. That's just so, I mean, I get goosebumps just thinking about it. So it's been really fun to plan that trial, and from that trial, we're going to get so much information about what it's doing systemically, and so we're collecting all kinds of data that will help us to understand how it's working in human patients. We have other studies that will look to optimize the geroprotection, and we're kind of taking a step back. So we'll go back to like C. elegans, which are like these tiny little worms that have a really rapid lifespan. So we can, and Drosophila, which are like little fruit flies, which are incredible model to work with because we know so much about the genetics of these fruit flies, right? So we can actually map not only the aging fly, which has been done with one of my colleagues here at Baylor, but then map the aging psilocybin fly and look at every single cell type in every organ and how it's changing with age and psilocybin. And then from those kinds of studies, we can better map the, what is the ideal geroprotective protocols, where are the safety ranges and safety limits, and sort of design better protocols that we can then further validate in mouse studies and beyond. And then we're also doing things like, given the protection we're seeing in these systemic organs. So, like, what if you give psilocybin in aging and then you have an injury model in mice? We do disease modeling, right? So, if we do that modeling after we've given the geroprotection, do they recover better? Do they have less severe injury? Do they have more recovery or repair of that injury? And so we're using the transcriptomic and proteomic profiling data to really look at, well, which diseases make the most sense to study. So lung is of interest. There are incredibly interesting protective effects with the skin. My colleague, Kosuke, who's my partner in crime on this project, he is really focused on the skin and wound healing angles. Even like cosmetic aspects of what it can do are really fascinating to us. We're also working on psilocybin derivatives. That's a really big focus of our lab right now as well. So, like, we have identified some psilocybin derivatives that don't cross the blood-brain barrier, meaning they don't get into your brain. So this is exciting for us because there may be people that do not want to have the hallucinations, but want the geroprotection, anti-aging benefits. Won't be as fun, but yeah, important for to be able to have other options, right? Where you don't. I mean, it's especially for elderly population. Now, what will be really interesting though is we don't know anything about the mind body axis, right? So maybe there won't be a protective without that. We don't know because we haven't put it in animals yet. So
That's exactly what I was going to ask you. I was going to say maybe there is a connection. You need to have that psychedelic experience in order to get the benefit of the.
totally, and we don't know the answer, but we're gonna find out. We're gonna find out really soon.
So that's actually an interesting. I know our time is getting shorter, but now we're getting into a more interesting place for me because the
Mhm.
science stuff is fascinating on its own, and I'm happy to have that now as our foundation. But what you just said about mind and body, like this research doesn't just concern itself with longevity and physicality. It's also, there's a spiritual component to this too. And I wonder, like, is that alive for you? Like, do you see yourself as like, yeah, you are a person of science and this is your primary interest and you have no curiosity about the spiritual stuff? Or would you say you're in both of those worlds?
Oh no, I am like fascinated by anything. So I mean, I'm just like voraciously reading everything I can all the time on all of it. And yeah, that aspect is really fascinating, and interesting to me. And I don't know what the results are going to be. I could come up with a guess for why it could go one way or the other, but I honestly don't know. I'm actually hoping somehow, I don't know why, that there is a mind body access connection, because I think from what I know about psilocybin, and the way that it's being used therapeutically is so profound, and it causes so much benefit for people. And so, yeah, I wonder if my guess, if I just like, because we haven't even started these studies yet, they're about to be started, but my guess is that maybe it will work, but maybe not as well. But I don't know.
Hmm.
I don't know. And at least in terms of its longevity benefits, but it's hard to say. And like when you think about like psilocybin and like when people take it recreationally, you know, there's what's legal and it's not. It's like decriminalized, and from a scientific standpoint, I never, I like to keep my opinions and things based on the science, because everyone's got an opinion about psilocybin, everyone. And what I want to be talking about is the science, because that's something really unique that, you know, for me, I can't argue with, right? But at the same time, I think there's a big difference from like a recreational use of psilocybin versus if it's administered in a therapeutic setting. It's very different. It's very different. And I think the
Yeah.
experience is going to be would be very different. And maybe the downstream effects would be very different because of that.
Yeah, and even within that spectrum of like the clinical conditions or environment, the recreational conditions or environment, and the ceremonial
Yeah.
conditions or environment are also very different. So I'd be curious what each of those different contextual circumstances, how it affects the outcomes. I mean, that'd be really difficult to study, I guess.
Yeah, it's a tougher thing to study. Well, I don't even know how you would study it, because obviously clinical trials, it's got to be given in like a certain setting, right? So really challenging to study something like that. But again, and this is just my guess, and I couldn't tell you why scientifically, but I just do think there's something to that. And I feel like I can see why therapeutic administration, where like, for example, like if people are taking it recreationally, I think typically, if they're taking it, they're out and about, they're with people. But when you're given it therapeutically, your eyes are closed, like you're in here, you're in your mind. There's no outside stimulation. Like, so I have to believe that the impact of that has got to be different than like when you're just taking it in a regular setting where there's people around and you're talking. And I don't know. But again, I don't know.
Yeah.
But you're right. It's like I can't even figure out how I would study something like that. But I think it's really interesting to kind of think about.
Yes. Yeah. And maybe one of the last questions I'll maybe it's not a question, it's an observation that came to me when you were speaking is that the times that I've ingested psilocybin and I've been in, say, nature
Mm.
and with people or without people, but this contrast to this more clinical setting where you're laying down, you have headphones on, you're just totally inward. When you're in nature, it feels almost as though you are communicating with this mycelial aspect of this creature, it's because it's a living thing, a fungus,
Mhm.
and how it works and it moves is like it makes a network out into the soil and it talks with trees and it does all these really interesting, fascinating things. And from a relational perspective, you could sense that when you're on it and you're in nature, you feel somehow connected to your surroundings in a different way than if I was in a room on a couch talking to a therapist. And
I
not
don't
to
know.
say,
I think people would say the same thing, and when they're given it therapeutically like that, believe it or not. That those are some of the things that I hear from reading about those studies. But again, yeah, I think that there's a lot to be learned actually about this, exactly the setting and how that works best, and what how do you design it so you can have the best impact. So much to learn.
yeah, it's amazing. And I wish I could talk to you all day about this stuff, but I want to respect your time. And
There-
maybe for a closing question, has this stuff changed your perspective of death, like your personal death and aging and longevity? What you've seen here is pretty fantastical, magical, surprising stuff. And I know more research needs to be done and, not get so excited, but for you personally, how has it affected the way that you see these things?
yeah, I mean, well, one is it's just like, I mean, it's blown the doors open for what I think anything I ever knew or thought about psychedelics, right? And just thought like it had psychedelic effects. That's it. I mean, I blew the door off that, right? Now it's just like, wow, this thing can, has these positive, benefits, but it can do so many other things. And maybe that they're feeding into each other. Like, like I said, that's why I'm really interested in this mind body access and how that works. And we actually have tools to test that, right? Because we have psilocybin derivatives. They're very potent, but they don't go to the brain. And actually, we're going to be able to at least scientifically kind of, maybe not to the extent that we're talking about here, like our kind of philosophical views and things, but we really will be able to use these things as tools to really kind of pick apart that mind body access and try to understand it better. So yeah, I mean, I think fundamentally, like, has it changed me? I mean, it's changed everything about the way I think about psychedelics. Literally had never even studied psychedelics. It wasn't even on my radar. And now I am like a full-blown psychedelics researcher. And we're looking at other psychedelics, by the way, too. So we actually are finding that, we're looking to see like how does psilocybin compare to other psychedelics, and that is also really interesting. So yeah, it's changed my life and my research world and my career. Yeah, I think when you study aging in general, you're like, I don't know about you, but like I'm almost 50, and I'm like now I've become like obsessed with like anti-aging, I've I'm more I'm more I've exercised more now. I eat better now than I ever have in my entire life. I don't drink. And psilocybin is a interesting, like, maybe part of my anti-aging regimen that will be, incorporated. So yeah, I think very differently about aging and longevity now that I am aging more than ever before. But when you're studying it too, it's just kind of like it's in the forefront of my brain all the time. So it's always on my mind. Yeah.
Thank you.
Yeah.
I guess one final question is, if people are interested in this and they want to find you, where
Mhm.
can they learn more? Where can they find you and keep up to date with what's going on in this field?
Yeah. Well, let's see. I have a LinkedIn page. They can always follow me. I always post kind of research updates and happenings on my LinkedIn page. And actually, it's funny because doing things like this, like podcasts, and people have heard them and contacted me. Like, for example, there was an athlete, an elderly gentleman that was, I think, in Australia, and he had heard a podcast and he contacted me, and he was showing me his like VO2 data and showing me that when was using his microdose protocol, he was doing like races, and his running performance and his VO2 max improved when he was on the microdosing. And then he showed me like these months I went away and I was out of town, and you could actually see the performance go down in those months. And so like things like this are cool to me because we're we are building these as exploratory endpoints in our clinical trials and mouse studies. And like I've surprisingly learned a lot from just people reaching out and saying, "Hey, I have this interesting thing I wanted to tell you about with psilocybin." Everyone's got cool psilocybin stories, but sometimes I'll gain insight from that and really build that into my studies. So yeah, feel free to reach out on LinkedIn. You can always search me. I currently work at Baylor College of Medicine in Houston, so you can always email me and get in contact. And I always love to hear from people.
Amazing. And thank you, Louise, for spending some time passing through with me today and sharing all of this amazing stuff. And congratulations on the human trials that are coming, and that your research contributed to all of that is incredible. And it sounds like it's been a wild ride. So I'm just grateful for your time and for you sharing your story here on the Passing Through podcast.
Oh, thanks so much. Thanks for having me. I mean, it's really exciting for me to be able to share this story because it just is such a wonderful story, and I'm fortunate to be the one living it. So, yeah, really, really, really grateful and fortunate. Thank you for having me.
Thank you for passing through today. Yeah, you, dear listener. Thank you for your support, for your attention. Thank you for passing through today, for spending some time with me and this episode's guest. If you're enjoying this content, please subscribe, rate it, like it, tell your friends about it, buy a billboard somewhere next to a highway promoting it. I mean, I'm kidding, of course, but your support really does mean a lot to me. So thank you for listening, and I will talk to you next time.